So let's talk about how the cell is supported and moves. We know that cells don't have bones and things like that, but they do have this thing called a cytoskeleton, and it gives them their shape. They have extracellular matrices that sort of hold them to other cells and kind of makes this sort of network of cells that work together. Cell junctions, how cells communicate with each other and how they kind of link to each other, and then cell walls. So let's go over some of these kind of slowly. A cytoskeleton. So let's first look at the picture. You can see that these are special dyes that outline the exterior part of the cell. This red is sort of giving the division point, that lipid bilayer of this individual cell. You'll see that this blue is the nucleus, and all of this green stuff is going to be different types of cytoskeleton. And they're usually these things called microtubules, and we'll talk about them in a second. But these are little, basically fibers inside the cell that give it its shape. And if some cells move, which some do, it'll actually help them with movement. But it basically tethers all of those organelles. These dyes, if we wanted to, we could use another dye for the Golgi apparatus and another one for the lysosome. But this one was really only just for nucleus, the microtubules, and then the lipid bilayer, just so you can get that. But there's other dyes that we can look at all the other parts of the cell if we wanted to. But what you'd find is that everything's kind of tethered to certain spots. And it does this because it is kind of like a bag, and if everything was just floating around, it'd be hard to get to. If they knew where it was and it was in a fixed place, it makes it easier to interact with. And there's three main types of fibers. There's microfilaments, intermediate filaments, and microtubules. And I put this list from smallest to biggest. The microfilaments are basically these intertwined little subunits. They're made up of these small things that could be built up and broke down. They're called actin. And they're involved in cell movement. So with muscles, they have this contrasting thing that they can make them expand and contrast. That's what makes the cells go really big and really small. And they do make themselves. So like, for instance, there might be a small fiber in one direction and it has to go in the other direction. These little balls come together to kind of extend that and sort of break itself down. The middle one is going to be the intermediate filament. And these guys are going to be rope-like. You can see it's got that spiral on it. It's reinforcing the cell shape. So this is the thing that gives it the shape, and it anchors itself to the organelles. So the organelles stay in one spot. These are definitely stronger. The microfilaments, they just kind of build up and break down, so they can go from cell movement and things like that. But these guys are really going to be kind of like the skeletal system of the cell. They're stable, they're permanent fixtures in the cell, where these guys are kind of temporary. They can get built up and then break down and go in different directions. These are definitely permanent structures of the cell. And then finally, we have these microtubules. And they're made up of these small components, these alpha and beta tubulins. And they help support organelles in the cell. Again, we have two different structures that support organelles in the cell. They drive things like motor proteins. So these are going to be like things designed for cell movement. Cilia and flagella. They're different things that are on the outside of some cells, and they allow them to go from point A to point B. So they're going to have to be a lot more stronger filaments, because they're going to almost be like, if you've seen a sperm cell, you've seen that big whip in the back. That's the flagella. And it's got to be a hardy, strong fiber to basically be able to spin and get that cell from point A to point B. They also help during that chromosome cell division. We're going to talk about that soon, where the cells will actually do this spindle fibers and divide the genetic material from one end of the cell to the other. They can disassemble and reassemble for that purpose. And they originate from the centromere in that process, where there's these small subunits that basically will divide the cell and then anchor to all these chromosomes and pull them from one direction to another. Talking about cell mobility, There's two different types. There's cilia, and what you're seeing here, this little slightly hair stuff, those are cilia. They're in the lining of our pharynx, so our pharynx is right below where our tongue is going down, and they're that tickle that every once in a while happens in the back of our throat. Basically what they are, they're constantly pulsing, and they're trying to catch flying debris, and they have some sticky material on them, mucus, where they can actually trap that material. If there's a lot of it, if it's between your nose and kind of like where your mouth sort of starts, it's going to make you sneeze it out. If it's from your mouth to kind of around where in a male has an Adam's apple, it's going to have you cough it out. And anything below that, it's gone into the lungs. So what's going to happen is it's going to use those little hairs to sort of kick it up as high as it can and eventually just drop it into the stomach to neutralize and kill those things. Here's the flagella. You can see this really long fiber. It's just one of them. And basically they're designed to move things from point A to point B. This will spin kind of like a boat motor and just make the cell go from point A to point B. It has to be a very strong fiber to do this. The similarities between these tubules are going to be a certain pattern. There's a 9-to-2 pattern, and what this really means is if you take this tubule and cut it in half, there's one structure, 2, 3, 4, 5, 6, 7, 8, 9 on the outside, and 2 on the inside. And we see this as a pretty common thing through all microtubules, that if we cut them in half, they always have this sort of arrangement from an animal to human to anything that really has this type of tubule. So that tells us that it's a flagellum, and they're all built sort of this way. The basal body also has its own arrangement. There's a 9 triple rings. You can see it's a little different on this side, and they're similar to those centrioles that pop up, and they're designed to split the cell. We'll see more about them in the future. We also talked about these extracellular matrices and what's on the outside of the cell. These extracellular matrices are stuff on the outside of the cell that holds the cells together. They're made up of these things called glycoproteins, which are basically some sugars and some proteins put together. And they hold the cell together, and then some of them are going to have specific functions. And then there's going to be another thing in the extracellular matrix called these integrins. These are proteins that span the membrane, so they go from inside the cell to outside the cell. And they can transmit even between one cytoskeleton to the next one and hold them together, kind of like two cans in a string, and they sort of have an interaction with the outside of the cell and the inside of the cell. So if we look at some of the stuff on the outside, you could see a couple things that we should be able to recognize. The lipid bilayer, this is the inside, the cytoplasm. This is the outside, this is the extracellular fluid. This long string is this collagen fiber. These are designed to basically hold tissue types together. You can see this connecting protein, and they connect cell A to cell B and just make sure that they're linked together at different spots. We have these integrins. They basically have a connection from the outside of the cell to the inside of the cell. And then the plasma membrane, which you've already talked about. And now you know what these microfilaments are. They're like the cytoskeleton of the cell. And in our next video, we'll talk about the 3 types of junctions.